Ah, yeah, thank you so much for waiting. I appreciate you being here so much. If I were giving this talk 10 years ago, I would have started by saying that the brain is the only thing on earth that can process language. And if we ever want machines that can process language, we're going to need to understand how the brain works first. This really was a common sentiment 10 years ago. Demis Hassabis, the co-founder of Google DeepMind, writes that the human brain is the only existing proof that general intelligence is even possible. But 10 years later, there's been a reversal. We have machines that can handle language just fine. Yet, we still lack an account of the principles underlying human language processing. What I'm here to argue today is that this is a good thing because all the techniques and methods that were successful in modern natural language processing, modern deep learning, can now be turned and applied to neuroscience as tools and lessons. So, this is a very broad thesis, and to give this talk some structure, I've broken things down according to the stages of the modern deep learning pipeline. And then, at each one of these stages, I'm going to talk about a project we're doing, an effort we're making to import advances from machine learning into advances in neuroscience. So I'll talk about data. I'll talk about how we collect data at scale to create the brain tree bank. I'll talk about modeling and how we use self-supervision to learn representations of brain activity for BrainBERT and population transformer. And I'll talk about how we evaluate those models, how we make evaluation more widely available with NeuroProbe, and how we do evaluation when evaluation gets a little tricky with BrainBits. Let's start with the data. And the question we're going to be answering in this section is, how do we collect brain data at scale? This will be a very fundamental part of the talk because the data we collect is going to be the sandbox in which all our modeling and evaluation happen. Datasets, especially large-scale datasets, have long been crucial to progress in NLP. Take, for example, the brain tree or pen tree bank, a collection of newspaper sentences from the Wall Street Journal, along with their accompanying constituency tree parses. Historically, very impactful because it was used in developing the first models that incorporated linguistic structure. But when it comes to the neuroscience of language, datasets, especially at this scale, really do not exist. To this end, we collect the brain tree bank. So the brain tree bank is a multimodal tree bank. It's sentences from movie dialogue accompanied by their audio and video tracks, and all aligned to patient brain data. This is joint work with Adam Yari, another PhD student in the group, and the purpose of the brain tree bank is for answering questions about language in the brain. Questions like, when and where is part of speech processed in the brain? And language processing happens at many timescales, and we want a recording modality that can capture all those timescales. So we collaborate with Boston Children's Hospital, and there are patients being treated for epilepsy. And a diagnostic part of this treatment is first inserting electrodes surgically, intracranially, and the neurologist watch the recordings from these electrodes to localize seizure onset zone, and in the case of surgical intervention, minimize downstream complication. But these electrodes are great. You can see them as bright spots on this CT scan. They directly record the electrophysiological activity of the brain at a high temporal resolution of 2 kilohertz and with a spatial receptive field on the order of tens of thousands of neurons. Really high quality data. Unfortunately, when it comes to neuroscience of language experiments involving this data, they're often very small. And this is unfortunate because it means they're small one-off experiments that each independently probe for a different aspect of language processing, and we're not often getting a picture of how all those pieces fit together. And The experiments are small by necessity. I mean, if you're a kid in the hospital and you have like a wire sticking out of your brain, you really don't want to sit through a lot of boring language experiments where you have to read a sentence one word off a time off a screen. And we thought to ourselves, okay, what do kids like doing? They like watching Marvel movies. So we show full-length Hollywood movies, and the patients watch as much as they want for as long as they want. And the idea is we're going to trade off control of the stimuli for volume of data. And then with enough data volume, we can go back and retrospectively subset it and answer any question we want. We like this because it, in a way, democratizes the neuroscience of language. This recording setup is very scarce and not a lot of groups have access to it. But if we can collect enough data up front, then other groups can use this data to do their own retrospective language studies. To make this possible, we need to extensively annotate the data. So we align the movie dialogue to the neural data at the millisecond level. Every word in the dialogue has its onset and offset marked by looking at the audio spectrogram, and every sentence in the movie dialogue is parsed into the universal dependency framework. This ends up being one of the largest universal dependency tree banks out there and the largest multimodal one. So already a very useful resource if you're interested in studying language and perception, even if you're not interested in the brain. But we are interested in the brain, and with high quality neural recordings, we can immediately see neural response to linguistic stimuli. So this is data from one patient, one movie, one example electrode in superior temporal gyrus, marked here in red, and each very thin row on this raster plot corresponds with one word. Blue is lower voltage as measured by the electrode, red is higher, and all the words have been aligned so that they start at word onset t equals 0, here marked by the dashed line. You can average over words and get a clear signal with low variance showing response to linguistic stimuli some 200, 300 milliseconds after word onset. And this is the type of temporally precise result and spatially precise result that you get because you have access to this intracranial data. One thing though, so this is a movie. There's a lot of things going on in a movie. There's explosions, and how do we know whether what we're seeing is a result of linguistic stimuli or something else, like brightness on the screen, or something the director is doing, or maybe like faces? To this end, we also annotate 16 visual, audio, word, and language features, and then we can use a generalized linear model, a regression, where the inputs are these 16 co-occurring features and the target is the mean neural response. And for this example electrode, we can interpret the beta coefficients fitted by that regression as the contribution of each feature to the response. And we see that for this example electrode, the linguistic features marked in blue, like surprisal, length, part of speech, index, and sentence, are the things driving responses for the electrode. But because we have data at scale, we don't just have to do GLM analysis. We can actually do direct subsetting studies. If, for example, you suspect that position in sentence modulates brain response, you can do something like take all the words at the beginning, middle, and end of a sentence, bucket them accordingly, average across words in each bucket, and indeed there is a differential response to words depending on where they are in a sentence. And you can subset this data further, controlling or balancing for other covariates. And when you do this across the brain, a picture emerges of what is being processed where. So here in blue, those electrodes for which position in sentence is a significant predictor, as assessed by the GLM analysis, and in white, the subset of electrodes that passes the more stringent control test that I was just describing. And because the data has such good temporal resolution, we can also do very cool decoding analysis. We can chop the activity up into very narrow time windows, train a decoder for each one of those time windows, and then what we get is a time course of decodability for this is a word onset detection task. And this looks superficially like the response curve I was showing earlier, but it shows something subtly different. This shows the point at which information about word onset first becomes available, when it first becomes decodable. And this is something that can be determined whether or not, the voltage literally makes a peak. Now, the type of decoding results you get are going to be dependent on the type of decoder you're using. And there's a trade-off. So on one hand, if you use a very simple decoder like a linear decoder, then your results are going to be very interpretable, but there's a high risk of false negatives because maybe the readings at that electrode are very noisy and it's hard to fit a linear decoder. On the other hand, if you use a very sophisticated decoder, there's going to be the risk of false positives. There's the risk that the decoder is so powerful that it's injecting information about the task via its prior. A solution suggests itself again from the world of deep learning. And the recipe for the solution that I'll describe across two models is we're going to do representation learning. We're going to learn good representations for the brain activity, and then we're going to decode those representations. And the question for this section becomes, how do we learn good representations? And that's what I'm going to describe with our model, BrainBERT. BrainBERT is inspired by what has been successful in the worlds of speech and audio modeling. And there, basically all of these state-of-the-art speech models today work according to this template. There are two stages. In the first stage, a pre-training stage, the model is trained over a large amount of unannotated, unlabeled data. And since there are no labels, we train using self-supervision. For example, masking parts of the audio spectrogram, and then the model needs to reconstruct what was masked. Then, in the second stage, the fine-tuning stage, the model learns to map the representations it has learned to English text. And then you can do something like automatic speech recognition with your trained model. And the intuition behind this staging is that in pre-training, the model is, you could say, learning the shape of language. It's learning phonemes and syllables and how they fit together. And in the fine-tuning stage, a comparatively smaller amount of labeled examples are needed than just to learn the mapping between the shapes that you learned and English text. We want to do the same thing with the brain, but we're not going to be working with audio spectrograms anymore. We're going to be working with brain data. And we play around with two different input representations. One, a straightforward short-time Fourier transform, for which during pre-training we mask constant width intervals of time and frequency, and a superlit representation. This is a composition of Morley wavelets for which there is a variable trade-off in the time and frequency resolution, such that at higher frequencies, where activity is more bursty or events are more bursty, there's more temporal resolution. And during pre-training, we use a masking strategy that reflects this. Basically, the amount of time information that's masked falls off as a function of frequency. And the goal of pre-training is to reconstruct what was masked. Fun little technical side note about training this model. So when we started, we found that the model got stuck in this local optima where it would just predict zero over and over because a lot of the spectrogram is zero. So what we found we needed to do was add this augmenting term to the objective, which basically says if there's a very bright spot on the spectrogram, if something's actually happening, make sure you reconstruct that well. And then we're able to start pre-training correctly. And all this pre-training, remember, is in service of something. It's in service of hopefully later on getting better downstream decoding performance when you do fine-tuning. And this is indeed what we see. So BrainBERT on this word onset detection task achieves 0.9 ROC AUC, outperforming the baseline decoders on the raw features, and red showing the example electrode where we do this decoding. And the critical thing about this plot is that not only does BrainBERT get better absolute decoding performance, it gets more data efficient decoding performance. So the x-axis is the number of labeled samples used during fine-tuning time, and for an equivalent amount of labeled examples, BrainBERT is getting better decoding performance. And I said we had a lot of data, but after subsetting and then subsetting again, really cuts down on the amount of labeled examples you have. It's like a cursive dimensionality thing. And always in the neuroscience regime, you're going to want your methods to be as data efficient as possible. So this is one example electrode. This is the decoding across the whole brain. And what's immediately apparent is the value of using the right decoder. So on the left, decoding from the raw features, and you can maybe get a vague sense of what is decodable where, but that picture only comes into sharp relief after you're getting decoding scores in the range of 0.7, 0.9, and you can really then localize with high confidence, okay, this word onset task is being done in these specific regions of superior temporal gyrus. Improving decoding is one thing. What really excites me about BrainBERT is it opens the door for other types of analyses that you can do. So I'll go back and forth drawing comparisons between machine learning and neuroscience. And one thing that people have observed for artificial neural networks is this trend in the dimensionality of its representations. So you look at the intrinsic dimensionality of the internals of a neural network, and you see that close to the inputs, the representations are very low. They expand in the middle layers and they compress closer to readout. And the working interpretation for this trend is that that expansion of intrinsic dimensionality corresponds with some sort of intermediate processing. And we might be curious and ask, is something similar happening in the brain? But it's not straightforward to compute the intrinsic dimensionality of the raw brain activity because the readings are noisy and it artificially inflates the estimation of the dimensionality. What you can do is compute the ID of the BrainBERT embeddings, which you can think of as a cleaned or denoised version of the signal. And when you do that, you find the regions with highest intrinsic dimensionality are in supramarginal gyrus and lateral orbitofrontal cortex. And these correspond respectively with phonological processing and sensory integration. And this makes sense for our movie task, but this is a preliminary result that we got a couple of years ago. I want to highlight work that our collaborator, Emily Chang, is currently doing at UPF Spain. And what she is doing is looking a lot more deeply into these trends in intrinsic dimensionality, in particular, the time evolution of dimensionality. And what she has found is that the time course of dimensionality here in black tracks with the time course of the decodability for a lot of these language tasks we were talking about here in orange. Just an example of result. She has many more experiments that she's going to be presenting this summer. There's one other thing that we think we can use BrainBERT for, and that's improving epilepsy treatment. So like I mentioned, these patients are originally in the hospital for epilepsy treatment, and the electrodes are recording their activity. The neurologists are reading the recordings, and what they're looking for is a characterization of the seizure network. Where is the seizure onset zone? Where do seizures start? How do they spread? Throughout relay paths, do they spread? How fast do they spread? And an important prerequisite task for characterizing this network is identifying precisely the seizure onset times. When do seizures start? So they're looking at recordings like this. They're trying to identify when we go from non-ictal or pre-ictal activity to ictal activity. And they're looking over like a week's worth of data because the patient is there for a week. And we think BrainBERT can make this more efficient because BrainBERT gives you the ability to represent intervals of activity as a single vector embedding. You can compute the similarity between embeddings, and you can group embeddings by similarity. And you can get then kind of bottom up a segmentation of the signal. And the inference we're going to make is that if there's a transition between pre-ictal and ictal activity, it's probably going to be at the transition points between one of these segments. This is also work that we've just started doing. It's a collaboration with Mass General Hospital. I'd like to highlight, I wonder why that's, okay, everyone's picture will be at the end. I'd like to highlight Grant Bell, the student at MGH who we're working closely with, and he did a lot of very cool supervised seizure onset detection work that's not on this slide. But I also want to recognize him because he biked all the data over from MGH to MIT. And it just turns out that for 10 terabytes of brain data, the fastest way to move it is someone with hard drives in their backpack, biking it over. Um, Preliminary result, what we're encouraged to see is that the transition points identified by BrainBird correspond more closely with, thank you, the annotations of the neurologist, at least compared to baseline like STFT features. Work that we're hoping to continue to do. Lots of people in the lab are involved in this. We wrote recently just 2 grant proposals for this earlier this week or earlier this month. And we have in the lab Vignesh and Abdul, who are going to be working on generative forecasting of this data to predict treatment outcomes. David and Shelley, who are going to be working on learning the segmentation end-to-end and doing event detection. And Grant and Mira and Niels, the domain experts in epilepsy who we're working closely with at the hospital. And on this slide, I have just as an example, the sort of chart that a neurologist would look at. One kind of salient meta property about this chart that hasn't appeared on many of the previous slides is there are a lot of time series here. We're not just recording from one channel, we're recording from many channels, and the neurologist needs to consider all of them at once. And BrainBird only gets you the representation for one channel. I had to do all of that analysis electrode-wise. And the question becomes, how do we represent multiple channels worth of activity? And this is what I'll address in describing our model of the population transformer. A similar problem actually arises in a different setting. So in video modeling, what was typical a couple of years ago was to have one model learn a representation over a single frame of data, and then have another model at another level learn to aggregate those representations into a movie level representation. And this is sort of the template of the thing we want to do with the brain data. It's a little more challenging because the electrodes, the sensors do not uniformly cover the brain, and the replacement varies widely from patient to patient. And this is the challenge we tried to address in designing the population transformer. This is work done jointly with the amazing Jilin Chao at Caltech. And the way the population transformer works is each electrode's worth of data is represented by BrainBird. So one BrainBird embedding per electrode, and we augment that embedding with the electrode's 3D position. All of this becomes input to yet another transformer, the population transformer. And this learns a representation for the ensemble of electrodes. Now, we want the model to be sensitive to 2 things. One, we want it to learn representations of individual channels in the context of its neighbors. So during pre-training, we randomly swap out individual electrodes with random activity, and then the objective of pre-training is to identify what was swapped out. We also want the model to learn representations for ensembles or groups of channels. And we simultaneously pre-train on an objective where the model is given 2 disjoint sets of electrodes and their corresponding activity. And the goal of pre-training is to determine whether or not these activities have appeared close or far apart in time. Now, again, all this pre-training is in service of getting better downstream decoding. And this is what we see. So we evaluate on 4 tasks, determining whether something is high or low pitch, high or low volume, the beginning of a sentence or not, or speech versus non-speech audio. And there are 2 things to pay attention to. So one is the green line, that's what we get with pre-training. One is the orange line, that's what we get without pre-training. And we've got the x-axis, which is the number of input channels. And 2 things that appear on every plot. One is that we get decoding performance that scales with the number of input channels. And this matters because it means it was worth going through all that trouble to model multiple input channels at once. The second is that with pre-training, we scale better. We get better decoding performance per input channel. And this means that all of the information about relationships between channels that was learned during pre-training ended up mattering to getting better decoding performance. Of course, we don't just care about better absolute decoding performance. We also want to check if we're getting more data efficient decoding, if we're able to learn more per labeled sample, if we're getting more compute efficient decoding, if we have to use less backpropagation steps to get equivalent performance. And this is an important one for any pre-training study. Do we get performance that scales with the amount of pre-training data? And we do. And this is so good because in these neuroscience experiments, we always have more unlabeled, unannotated data than we have annotated data. So any decoding approach that scales as a function of the amount of unlabeled data is a huge win. Of course, like BrainBird, we care more just about we care about more than just improving decoding performance. We also want to see if we can learn something about neuroscience. And one thing that neuroscientists like to do is they like to build connectivity maps of the brain. How is one region functionally connected to another region? And this is usually done by computing the cross correlation between regions at resting state activity. But now that we have a model that learns contextual representations of individual channels, in the context of its neighboring channels, we have another way of getting at this information. What we do is we hold out individually single channels from the input, and then on the output side, we measure how performance on the pre-training task degrades. And the inference we're going to make is that if another channel has its performance greatly perturbed, then there was a relationship between those channels. One channel was very useful for predicting what's happening at the other channel. So we do this for one channel at a time. We check all pairs. We can average across all pairs, and we get a map of how regions are connected. What's really cool about this is we do recover the edges found by the traditional connectivity analysis. We actually get a superset of the edges, and this is because the population transformer is going to be sensitive to relationships outside of cross correlation, outside of whether or not the waveforms literally overlap. Okay, that was PopT, BrainBird, how we do decoding. One thing that I think hasn't been apparent from the slides is how much custom code we had to write in the pre-LLM days to get the evaluation working for these models. Braintree Bank is not a benchmark, it's a data set, and we had to do a lot of finagling to get the tasks right, get the splits consistent, and this is not ideal. Ideally, our evaluation should be reproducible. In the best case, it would be so reproducible and so easy to use that anyone could pick it up, download it, and evaluate their own models. And if it's easy enough to use, it might on its own start spurring the development of other foundation models, because a lot of the times the community is waiting on a benchmark to start measuring against before it really takes off and new methods start getting proposed. How do we make our evaluation more widely available? Well, this is the question we try to answer when we put together NeuroProbe. NeuroProbe is a joint work with Andrei Zaharodny, who is now at Merge Labs, and Jilin Chao from Caltech. And I really have to give credit to Andrei for growing this project. He started it out of the evaluation he was doing for his own foundation models. And what we do is we take the evaluation we were doing for BrainBird and PopT, and we standardize it. And we also add a couple new challenging settings. So in our original evaluation, we were training on one portion of a session and testing on another portion. With NeuroProbe, we also have a setting where we train on one session, test on another session, and train on one subject and test on another subject. And you can kind of see by the decreasing performance of the models that the settings are getting more and more challenging. We expand the set of tasks from our original 4 to 15 audio, vision, and language tasks. And what you get then is a profile of each model's decoding ability that you can use to rank models against each other. And my favorite thing about this plot is that the best model is not PopT or BrainBird or anything we developed. It's actually Diver 1, a model from a South Korean team. And NeuroProbe is still in preprint, but Andrei has been so good about advertising it that we're starting to get submissions from other universities to our leaderboard. And this is really an area where we want to be beat. We want better and better submissions to come to our leaderboard and beat us and get better and better decoding. Of course, okay, I'll keep returning to this. We care about more than just getting better decoding. We also care about learning something about the brain. And now with 15 tasks, what we can do is get that full picture I was describing when I was talking about Braintree Bank of the spatial distribution of where all these different interrelated multimodal language processing tasks happen in the brain. So you can immediately see things like maybe audio tasks like volume and pitch are most decodable closer to audio regions in superior temporal gyrus, temporal lobe, and more linguistic tasks like speech and sentence onset are more decodable in a wider footprint that includes those audio areas and some of the frontal lobe as well. And these vision tasks like optical flow are more decodable closer to the back of the brain, occipital lobe, closer to those earlier vision areas. And we can blow this chart up and get the time evolution of each task. So this is sentence onset decoding, and you can see that in early, like close to the beginning of the sentence, this task is most decodable, close to those audio areas in temporal lobe. And as time goes on, it becomes more decodable later in the frontal lobe, maybe where something more abstract is happening. And just for effect, I like to show the time evolution of all these tasks playing at once as a movie. That went really fast, so I'll play it again. I like this a lot because it's a movie of what happens in your brain when you watch movies, and we're watching it as a movie. I'll come back to it if it ended up being too fast. One other thing I want to say about benchmarking is there are many ways to do benchmarking. So you can benchmark based on decoding ability. You can also benchmark based on how similar the brain is to a model. And what I mean by that is you've got the brain, you've got brain activity, and you've got the internal representations of a model. And you can measure the representational similarity between these two things. And this opens up a new experimental framework. What you can do is you can take a model that is trained for a particular task, and then you can check where in the brain its representations best align. And then the inference you can make is wherever it best aligns, there the brain is doing either that task the model is doing, or it's doing something similar necessary for that task. This is a framework pioneered by Jim DeCarlo's group across the street and Martin Shrimp. And they have done really cool work in identifying like language processing and aspects of language processing in the brain using this framework. We use it to answer a different question. We use it to ask where in the brain is multimodal integration happening? So by multimodal integration, I mean some synthesis of vision and language information. And what we're going to do is we're going to take models that are trained only on vision, only on language, and we're going to check where they best align with the brain. And then we're going to take models that do some nonlinear combination of vision and language, and we're going to check where they best align in the brain. And the inference we're going to make is wherever these multimodal models are better aligned than any of the unimodal models, that's probably where multimodal integration is happening. Okay, this is work really led by Vignesh Subramaniam. It was a couple years ago when we did this experiment, and I highlight it here because it's so cool. Okay, it turns out that we actually don't have control of electrode placement. This is all determined by clinical considerations. So the visual areas of the brain are super undersampled, but I would say we get very suggestive results. We get candidate regions in temporal lobe and maybe like closer to those visual regions where follow-up, I would say, is warranted on determining whether multimodal integration is happening. And then we can also rank models, which we love doing. We can now, for each multimodal candidate region that we identify, find the model that best aligns with activity at that region. And we get a ranking again, but now this isn't a ranking determined by how good decoding performance is. This is a ranking of how similar the brain is, or how similar the brain is, yeah, to that model. Okay, when it comes to classification and representational alignment, like we were just talking about, reporting progress is very straightforward. Just give the accuracy or the correlation. But when it comes to more complicated decoding, like trying to recover the image entirely that a patient was looking at, evaluation becomes more tricky. And I'm going to talk about how we evaluate generative reconstruction methods. And this will be the last project I talk about today. It's tricky because I would say the models have gotten too good. So this is an image shown to a patient in an fMRI, and this is a reconstruction predicted by a model that only has access to that patient's fMRI data. And I would say this looks fantastic. It blew me away when I saw it. And just this was a couple years ago. And every year since then, the images have gotten better and better. And on the face of it, the takeaway would be, oh my gosh, we're getting so good at understanding visual processing in the brain. But if you dig into each paper, what you see is that a lot of the improvements come from swapping this year's image diffusion model out for last year's. And you start to wonder, are we better, like getting better at understanding the brain, or are we just getting better at generating images? And this is the question we ask with BrainBits. This is work done jointly with fellow PhD student David Mayo and Asa Harbin from Lincoln Labs. And what we propose doing is inserting an information bottleneck into the reconstruction pipeline. And then we can tune the width of this bottleneck. And what we're looking for is the point at which reconstruction performance plateaus. And the inference we're going to make is that past that point, the model is no longer extracting useful information from the data. So that minimum bottleneck width is kind of the ceiling on the model's ability to utilize brain data. And I'll just jump to the punchline. You can get the input down to a very small size and still get what I would call reasonable reconstructions. So here are the reconstructions on the left for each one of the different bottleneck sizes, 1, 5, 10, 25, 50, and then on the right, the original reconstruction. Here's one example. Here's a couple more. Take a look at the second row. It's supposed to be a plane in the sky, and I would say with 10 floating point numbers, so down from 15,000 input voxels, you have most of the main semantic elements. You've got the sky, you've got something in the sky, and you can kind of eyeball it. I'd be curious to know after where you guys would draw the line. But you can also do this quantitatively using the metrics that the original reconstruction methods use and see that indeed somewhere between 10 and 20 performance plateaus as a function of that information available from the brain. And the takeaway I'll end on is, okay, getting good images is all well and good if we care about getting good images. But if we want to measure the progress we're making towards what I would call the harder problem, towards our understanding of the human visual processing system, then we need to care about more than just image quality. Okay, that was all the projects. I'm going to do a recap, but let's recap in reverse. So we just talked about BrainBits, which is how we evaluate generative reconstruction methods. We talked about NeuroProbe, which is how we do evaluation and make it more widely available. We want evaluation to be more widely available because we want to spur the community's development of better and better foundation models, models like Population Transformer, like BrainBERT. And we want better foundation models because we want better, more data efficient, more accurate decoding on language and the brain data, data that we hope to collect at greater and greater scales. And I'll keep going in reverse because I want to end on a wild prediction. We talked about how we're starting to use machine learning to get advances in neuroscience. And as we start to get better models, as we start to get better recording modalities like single neuron recordings, as we start to get more coverage of key areas of the brain, I predict in 10 years, we could end up in a position where we have intelligent systems just by virtue of recapitulating the brain at the level of recorded activity. And this is not the old dream. So the old dream was to understand the brain so well that you could, like with pencil and paper, write down the algorithm it was performing and then implement that algorithm in a machine. This is not the new approach either, or it's not the current approach, which is training a model on inputs and outputs. If this ends up happening, it would be, I'd say, a new pathway to intelligence, and I would say would bring things really full circle. Okay, thank you. Okay, thank you so much. I want to do questions, but first I want to say thank you to a couple of people. I need to say thank you and recognize all my brilliant, hardworking, very smart colleagues, co-authors, and collaborators who all had a very important hand in this work and got results or like feedback or gave ideas. And I want to especially thank all fellow members of the InfoLab. I'd say it's really been a joy working with you over the course of the PhD. I'm so lucky to have had a place where I was excited to go into work every day because I knew I was going to do interesting projects with smart people and have like great conversation. And yeah, it really was a pleasure. I want to thank, of course, my committee, Philip and Paulina and Boris, for the early hand they had in shaping this thesis and all their time and advice. And I want to thank the three people who had the most time spent mentoring me during this PhD. So this is my PIs, Boris and Andre and Brian. And I am so fortunate to have had uh, advisors that care so deeply about their students and invest so much. And you can see definitely that they put a lot of effort into each individual student's growth. And I've learned so much science and about how to do science, but I've also learned so much about like the scientist that I want to be. And I credit Boris and Andre and Brian for creating a really amazing lab culture that is very productive, but also feels very supportive. And okay, I'll just do like one illustration of the way Boris does things. So I don't think Boris ever told me to come back into the office after the pandemic, but he has like a thing of chocolates on his desk and a coffee machine in his office. And then if you come by, you get a chocolate. And sometimes he brings the chocolate to you. And and then people just end up being in the lab a lot. Uh, yeah, I'm so lucky to have worked in the Info Lab these past 5 years. And thank you, Boris, Andre, and Brian for really making the Info Lab a special place. Um, okay, I would like to thank family, family from Weston. It has been so good to have lived close by all these years and to have seen so many of my cousins' amazing plays. I want to recognize my grandmother, who is here at MIT today and was here at MIT over 60 years ago studying civil engineering. Oh, I guess you never really can escape the Institute. I want to thank, oh, I did this in the wrong order. Okay. I want to thank all the amazing friends from Oklahoma, the OGs from a long time ago. I want to thank friends from Cambridge. I'm so fortunate to have known so many smart, fun, kind people. Yeah. Thank you so much for making dinners, bringing snacks. Oh my gosh. Taking time to be here in the middle of a random workday. I appreciate you all so much. Um, you know what they say, word embedding is only as good as its K nearest neighbors. And okay. Okay, thank you. I wasn't sure if I should have said that one. And I feel very fortunate to have been uplifted by neighborhoods of such high quality. Um, Finally, I want to thank and honor my parents, my mom and dad, and my sisters who flew all the way from Oklahoma and California to be here today and who woke up earlier than me to buy sandwiches for the room. Yeah, thank you so much for the unending support and encouragement from the very beginning. Thank you for making, I'd say, all of it possible, and thank you for making it worth it. Okay, thank you, and the audience for making it to the end with me. I really appreciate the time. Let's keep talking about the brain. I'm happy to take any questions. Okay, yes.
Could you explain exactly what like ROCAUC is when you're talking about like linear
Oh, yes.
process?
It is the area under the receiver operated curve. So when we're doing the classification, oh yeah. Um, yeah, what is ROC AUC? Basically, when we do the classification, we actually don't set a threshold for what's considered the positive or negative class, but we can vary the threshold and then look at the balance of like true positives and false positives across many thresholds, aggregate them together, and that's kind of like the average accuracy across many possible thresholds.
Yeah, I wanted to ask more about the very last slide you had where you're speculating on a future training paradigm being, suppose you have a learning model that is given a large corpus of brain patterns and suppose it learns faster from these brain patterns. So your work on, for example, in brain fits, right? You saw that the inherent rank of the human mind wasn't very high. So I guess the question I'm asking you to speculate on is, do you think that models which learn in this way, where you're kind of, I don't know, next token predicting the human brain state, do you think that are going to learn things in a more efficient manner, or are you speculating that due to, our schemas and our brain being a product of evolution and not so much training loss, we end up learning more garbage rather than something that's more minimal and passable on?
Yeah, okay, I want to make sure I get this right. I think one thing I'm hearing in that is how did history work out this way that we have models that work the way they do and don't work like the brain? If one of them like really is more like minimal or like efficient from like evolution standpoint, why did our machines ending up looking the way they do? Or
It's more, I mean, yeah, that was the backbone behind the question, but more of, as you've shown in brain bits, our brains are not so super efficient. I'm assuming if you use like a standard encoder, you can even get the rank lower, right? Because we're not dealing with whatever noise or crap is in the representation of our brains, right? So, I guess if you could speculate on that remark, I just made your speculation on how learning would happen from predicting brain states.
yeah, I think, okay, so brain bit seems to indicate that we have, or the brain has many redundancies that are probably not worth reproducing. Yeah, I would agree. I mean, evolution probably gave our brains a lot that are not going to end up being needed for something that's like learn end to end, straight towards the task. I would say the fact that we're able to compress so much information about the brain and still get the same results of the reconstruction pipeline says more about the task than what the brain is doing, though. Task is very simple and very stereotyped. So the other piece I didn't mention about this reconstruction task is I kind of consider it not to be reconstruction, but more like multi-way classification. There are only a few, or like not a few, there are a finite number of classes in the COCO data set, which is what the patients were seeing from. And most of the reconstruction ability, I think, comes from the model just being able to get the right semantic category. And then from there, the generative model can do a lot of the heavy lifting for filling in the pixel level information. I'd say there's a lot in the brain that we should not take for granted because that particular task can be compressed so much. Oh, yes.
So sticking neurons or electrodes in someone's brain is very invasive. And then you showed later a less invasive technology, fMRI. What do you think the limit is for like how simple the reading technology could be to get the kinds of information that you're going after? And part of my question is like, how soon will someone be able to literally read my brain from the outside of the house?
Okay. Oh, this is so great. First part, I think it needs to be intracranial, or there are so many great or just like without getting into what people have found they can do with intracranial data, just going by the stats, the intracranial data has so much that all the other recording modalities do not. So it's not distorted by, the skull like MEG or EEG is. It's very spatially precise, which MEG and EEG are not. And it's also very, temporally precise, which fMRI is not. And it's even it's not the best recording modality that exists out there. There are like neuropixels or like single neuron recordings that you can do intracranially, and um, anything besides that is a lot more difficult to get, or like it's a lot higher signal to noise. The fMRI, the EEG, and that's just going by the stats and the studies that people do. All the really cool stuff, for example, that like Eddie Chang does out in California, where he's like getting music or like language out of people's heads. It's all intracranial. And um, I think if there's any hope of ever getting thoughts out of people's heads, it'll have to be um, through the skull. Okay, so the second part, how close are we to reading people's minds? Um, there are So people are on this. We did not do this, but there are people out there who have what they're called like imagined text decoding, like benchmark, and they put people in these recording setups and just like think really hard to yourself. And probably so the main barrier to getting that technology is getting a data set that's reliable enough. And then the other thing is the coverage. So we were lucky because the people in the epilepsy monitoring unit were actually what they cared a lot about was not interfering with the patient's language ability in the case of surgical intervention. So it just happened that they were really heavily sampling the language areas. Um, but um, that's for like language production. For the thoughts or like wherever thoughts may land in the brain, they were not sampling for that. And I think that the studies that I just mentioned are also in a similar position. Uh, it would be a very special set of circumstances that delivered us sampling coverage in those areas.
Among your set of tasks, there's one labeled GPT survival. What is that? Maybe specifically among those, like some are easier and harder than others. Intuitively get an idea of like what makes something easier and harder for the models to be more on.
Yes, yes, yes. Okay. GPT surprise. All right. So we put that in there because we had a lot of word level features and we wanted some like the next level, like semantic level features in there. And GPT surprisal is what would a well-trained model deem to be surprising in a sentence? Or so surprisal is what it's called, but it's actually like the uh, like log probability of this next token given all the previous tokens. And we had a couple studies that we wanted to run where it's like basically does the machine find the same word surprising that a person would? And to a degree, yes, those two things are correlated, like seeing the GLMs that we were fitting, but not to the same degree that some of the other tasks were. This was one of the more difficult decoding tasks. I would say in general, the more difficult decoding tasks ended up being the okay, like if there's like a scale of like concrete to abstract, they ended up being the abstract ones, which we really wanted them to be, like, we wanted them to be decodable. And we just found that it was very difficult to see things as the features got more and more abstract, like part of speech or like Uh, we have also like grammatical role annotated in there that didn't end up making it into the final benchmark. Yeah. Oh, yeah.
You're probably more, you get the attentions between the different neurons, right? Do you think there's anything interesting we could go in by looking at those and seeing how the different areas of the brain, like going, different level of understanding path from one to the other, depending on, like the word or the context of the feature?
Wait, this is such a good question. Okay, we actually ran this analysis. We have, okay. So yeah, you've got it right on. We have inputs from the brain, and it's a transformer, so it learns attention maps over these inputs, and we can, for the 4 tasks we evaluate on, see what matters as far as the attention weights are concerned in the brain to doing this prediction. And we do find something really cool, which is that for the speech tasks, like speech versus non-speech, we get a lot of attention concentrated on, this back part of the temporal lobe, or like Wernicke's areas, or like the area that's been associated the most with speech. Um, there's there's a lot of trickiness when it comes to evaluating attention weights. Attention weights put a lot of weight on like non-informative tokens just as an artifact of the noise in their training. We didn't do anything to clean for those artifacts, but yeah, if we were going to go back and do this again at like a much more focused level, I think attention maps are definitely worth a closer look. Oh, okay, wait, this is really good too. That was for language. We're doing stuff with seizures now. And okay, all of the like cleanup we want to do, we're going to apply for the attention maps we're going to be building for our seizure detectors. And that's another area where we care about getting interpretable features out of the model. Oh, yes.
First of all, compliments for covering so much ground for the president. My question for that is that, so if you have focus on one thing and you had unlimited resource to everything, what would you do with the next one?
Okay. I was so lucky to have, be in a group that has access to this recording setup and has these amazing collaborations. Okay, so 2 things that I've always really wanted to try, and if I had like 5 years infinite funding that I would do, one is like the motor stuff. So all of this is language. I think there's a ton of really cool work being done where, they are sampling the motor areas and they're getting very cool like interface between like robotic arms and like the human brain. And I would really like to take a crack at that. And the second is I really want to push that wild prediction that I ended on. I think the buzzword most associated with that prediction would be like digital twins, or we're collecting enough data now at enough scales that I think a digital twin could be like not a crazy thing to attempt, or a model that like forecasts future activity from previous activity and tries to mimic the brain's outputs given its inputs. Um, I would like to spend a couple of years just gathering data from, they're like spread out across many different data sets and getting them into one like a forecasting training pipeline.
This person, I'm an anthropologist who is curious about how science and technology can impact the world, like outside of academia. And I am curious with the research that you've done here, how do you, I mean, it's kind of a speculative and not a question that is part of the evaluation. How do you think it can impact people 5 or 10 years later outside of academia? You mentioned, treatment for epilepsy and children, but yeah, for sort of the brighter and stuff, darker.
Okay, this one will need some thinking. Thank you. I'll do where I see it going for neuroscience, and then I'll do like more broadly. Of course, the neuroscience, I see this having the potential to make treatments a lot, or like diagnostics a lot more efficient. A lot of, the diagnostics we do these days rely on the manual labor of highly trained experts, and just a lot of the first filtering layer of the work they do takes a lot of time. And I see a lot of potential for these methods to make that go quicker so that they can put their eyes on the most relevant things faster. The maybe there's like echoes of the earlier question about like how close are we to reading people's mind. I actually think that part of the application is a long way down the road. Just because I mean I have a hard time knowing what I'm thinking all of the time, and I have access to all of my brain's internals, and um, uh, I I see potential in it being used for um, interfaces like um, things like Neuralink, where maybe no interpretation is being done on the features, but in a feedback loop, the human can kind of like get better at using it. So not necessarily like it understanding the brain more, but the human gets better at using this technology that, is being exposed to some of its neural activity. I see being very possible. Or, I mean, indeed, it's happening. It's kind of happening now. And more broadly, so outside of neuroscience, I think a lot of these problems that we try to solve end up being applicable in many other areas as well. Brain data is time series data, and anomaly detection and clustering and semantic segmentation are all things that are going to matter wherever there's a complex, like multi-channel system, finance or like earth science, or very many related areas, I think are going to be able to make use of some of the approaches we've developed here.